The profound shift of the psychedelic experience begins the exact moment a molecule successfully binds to the serotonin 5-HT2A receptor. Neuroscientists typically map these receptors within the brain's cortex, specifically on the large pyramidal neurons responsible for high-level processing. When these receptors fire in the brain, they trigger the excitatory signaling that leads to altered perception and visual hallucination. However, the same 5-HT2A receptors are also located outside the brain. They exist in high densities on the lining of our blood vessels and on the immune cells circulating through the body. On an immune cell, 5-HT2A activation performs a different role. It initiates a signal that suppresses the cellular machinery of inflammation. The 5-HT2A receptor functions as both a gateway to altered consciousness and a primary regulator of the body's immune response. Current psychiatric research often credits the success of psychedelic therapy to psychological breakthroughs or the growth of new neural connections. Clinical papers focus on the subjective quality of the trip as the primary driver of patient recovery. This psychological model overlooks the systemic physiological changes occurring in the body, specifically the modulation of the immune system. Laboratory measurements show that activating the 5-HT2A receptor produces an intense anti-inflammatory response. This physical suppression occurs at concentrations far lower than the threshold required to trigger a hallucination. By analyzing the in vivo data and cellular mechanics, we can see how these compounds treat depression through direct physical intervention. Modern medicine is now beginning to account for the physical anti-inflammatory mechanisms that have previously been overshadowed by the psychedelic experience. In 2013, the Charles Nichols lab published a study on the effects of a research psychedelic called RDOI. The study monitored TNF-alpha, a protein that acts as the primary driver of systemic inflammation throughout the human body. Researchers administered RDOI to animal models to test its ability to stop inflammation in the gut and the vascular system. As RDOI concentration increases along the x-axis, it blocks TNF-alpha and forces pro-inflammatory gene expression toward zero. The compound specifically deactivated IL-6, ICAM1, and VCAM1 genes. This complete blockade occurred at infinitesimal picomolar levels. At this concentration, the compound matches the potency of the most effective anti-inflammatory medications measured in laboratory settings. These results were consistent across aggrethrin vascular systems of the test subjects, proving the effect was systemic rather than localized to a single tissue. A molecule primarily known for its psychological effects demonstrated the ability to outperform standard anti-inflammatories at a cellular level. This evidence leads to a practical question. Does the body require a full psychedelic experience to receive these immune benefits? We can find the answer by comparing the dose-response curves. While the immune response happens at a low concentration, the curve for behavioral and psychedelic effects only appears at a much higher dose. There is a significant mathematical gap between these two events. Inflammatory suppression is achieved at a fraction of the dose needed to alter a patient's consciousness. This separation proves that the 5-HT2A immune mechanism functions independently of the subjective psychedelic trick. It is possible to activate the physical healing properties of this receptor without ever reaching the threshold of altered perception. These immune-regulating effects also take place within the central nervous system. Inside the brain, resident immune cells called microglia govern the inflammatory environment. Overactive microglia cause neuroinflammation, a condition driving Alzheimer's and Parkinson's. Tryptamines, like psilocybin, bind to microglia receptors, signaling the cell to change behavior. This reduces the production of tissue-damaging pro-inflammatory cytokines. Instead, the interaction triggers the release of IL-10, a protein that actively cools the neuroinflammatory environment. By reducing this neurotoxic stress, psychedelics establish the chemical stability necessary for new neural connections to form and survive. This biological data aligns with a shift in how psychiatry views major depressive disorder. Researchers now categorize many cases of depression as systemic inflammatory conditions rather than simple neurotransmitter imbalances. High levels of inflammation in the body are closely linked to the physical symptoms of depression, such as chronic fatigue and the loss of interest in activities. 5-HT2A agonists address these symptoms from two directions. They promote neuroplasticity
while simultaneously cooling systemic and local inflammation. These compounds are effective against difficult-to-treat depression because they treat the mind by directly addressing the inflammation in the body. The reach of the 5-HT2A receptor extends into many areas of physical medicine outside of mental health. Because the anti-inflammatory effect occurs at subperceptual doses, it has become a primary focus for pharmaceutical development. Laboratories are now designing molecules to target the 5-HT2A receptor specifically for chronic conditions such as asthma, atherosclerosis, and Crohn's disease. The objective is to utilize the receptor's ability to suppress TNF-alpha without producing any psychedelic effects. The discovery of a reliable biological regulator for human inflammation may be the most significant result of the current era of psychedelic research.